All right, last lecture. This one will be shorter, I promise. We're going to talk about nucleic acids. What the heck are these things? What is a nucleic acid? Nucleic acid is stuff like DNA. Maybe you've heard of it. RNA. We're also going to talk about one other type. There's a couple others, but ATP. Okay. What is DNA? What is RNA? What is all this? I don't have a little checklist here, but I'm just going to keep cruising. And we'll go through these. So DNA is deoxyribonucleic nucleic acid. DNA, deoxyribose nucleic acid. And then RNA is ribonucleic acid. Deoxyribonucleic. This is actually all one word. Sorry, I knew I was not getting that right. It's like this: ribonucleic acid, and then this is deoxyribonucleic. I told you I'm terrible at spelling. Why can I not select that? Okay, ribonucleic acid. Deoxyribonucleic acid. I know you guys have heard of this. What is this? Well, in very general terms, it looks like this. It is a double-stranded, what we call a helix. And it's got little rungs in the center, something like this. It's like a little ladder. You may have actually heard it referred to as the DNA ladder. Ribonucleic acid looks something like this. Rather than it being two strands like this guy over here, it's just a single strand. Pretty cool. Those are the two main differences. There's other little differences, and we'll talk about those. So what is a nucleic acid? These are nucleic acids. A nucleotide, this is actually technically a nucleotide. It's a specialized nucleotide. These are nucleic acids. Nucleic acids. And this is a nucleotide. Nucleotide. Okay? These are made out of nucleotides. So nucleic acids are made out of nucleotides. Nucleotides make up nucleic acids. Just like amino acids made up proteins. And fat, I didn't talk about that, but a fatty acid, we should put this back in your notes. I didn't talk about that. But these little tails in a triglyceride, this is called a fatty acid. Everything's made out of acids, it turns out. That's a fatty acid. They make up triglycerides, a fatty acid. These would be also fatty acid. Okay? All right, back to business. So nucleotides, what is a nucleotide? Let's just draw one, and this is going to be the only one that we draw. So on the butt here, we've got a polar region, just like a phospholipid, actually. We got this little polar head, and we'll kind of draw out what that looks like just so you can get an idea of it. Then we got some carbon going here. Carbon, carbon. And then actually down here we got a sugar. It's a 5-carbon sugar. Looks like a little house. That sticks like that. And then coming up here, we've got another 5-carbon sugar. Looking kind of thing, but this actually is not a sugar because it's got a bunch of nitrogens in it. But it kind of looks like a sugar. It looks like a similar thing. It's a sideways house. But it's got nitrogens in these rings as well. And then we got a little 6, like so. Okay, I should have drawn these in different colors. I'll highlight this maybe. looks like trash. This looks like garbage. Let me just draw them in different colors. It won't take that long. I'll draw these in that color. Nope, I want the pencil. Okay. Last one's of course gonna be better because now I'm gonna do it faster, sloppier. Bang, bang, bang. Okay, so this part here, it's got phosphate in it. Remember, those are very electronegative. It's also got oxygens all the way around that. That's called a very specific kind of thing. It's called a phosphate group. So these would all be oxygens also. And there's a bunch of bonds in here. I'm not going to draw them all out. So this thing is very negatively charged. So it likes to interact with water. All right, and then this down here would be a pentose sugar. Pentose meaning 5, pentose sugar. And then this over here is a nitrogen base. So this forms the backbone. This forms this part of DNA. This phosphate group likes to interact with water. It's polar. And then inside we got this nitrogenous base. That's these things in the middle. And if we were to display this out and make it straight, it would look something like this. I'm going to redraw this in pink because I drew it in pink there. So if we were to draw this as a very just like straight ladder like this, it would look something like that. And we got different types of bases. And you may have heard of some of these. I'll list them out. I just want to make sure I spell all these right here. So for DNA, we've got 4 bases. Got adenine, thymine, guanine, and cytosine. For ribonucleic acid, we got all the same ones. So I'm just going to copy paste that for RNA, except our thymine changes to a different one. We have uracil. So this is the only one that's different. We'll highlight that one. Uracil is different. Okay? All right, so in these bases, in the center here, we've got that pentose sugar, but then there's potential for these different bases. So for example, we got our pentose sugar. I'm not going to draw them all out, but well, that's not what I want. I'm just going to draw like 1. Nope, that's not what I want. 1, 2, 3, 4, 5. That seems good. 1, 2, 3, 4, 5. Okay, and as a rule, we'll do an A over here. And then maybe we'll do another A. Whoops. That's not what I wanted. We'll do another A. And then another A. And then in between, we'll do, let's say, some G's. I'm a G. Oh, I don't know what's going on there. We'll do some G's. We'll do a G there. And then we'll do a C there, let's say. Okay, so A always binds with T, and T always binds with A. G always binds with C, and C always binds with G. So I'll write that over here. A, do it in black. A always binds to T. G always binds to C. All right, so over here we need a T. We'll do those in this. This blue, no, it's too close. We'll do those in orange. We got T, T, T. And we got G. And we got C. Okay? So that these bases in between the DNA actually zip the ladder up together. And the way they do that is with hydrogen bonds. If you remember what a hydrogen bond is, we'll go back to water here. So water has this polarity, this semi-charge, because electrons like to spend more time with oxygen than they do with hydrogen. So hydrogen's slightly positive. So if hydrogen interacts with something that is slightly negative, for example, oxygen, it'll create a hydrogen bond. It's created by the partial charge across the molecule, meaning that there's this magnetic charge now between the hydrogens of one water molecule is attracted towards the oxygen of another molecule because the oxygen is slightly negative. It's funny how we keep coming back to that. Look how important water is. It makes these hydrogen bonds. And oxygen and hydrogen can do that in other molecules, such as in nucleotides. So A and T will have hydrogen bonds between them. I'm not going to draw out the details of it, but there will be 2 hydrogen bonds. So I'm just going to draw those as like little lines. So A and T are going to have 2 hydrogen bonds with each other through their oxygens and hydrogens. Oh, what did I do there? That should be a C. How come nobody corrected me? That should be a C. Guanine and cytosine have 3 hydrogen bonds. So they're actually a little stronger. A and T have 2 hydrogen bonds. And finally, oh no, we got a couple more. So C and G have 3 hydrogen bonds. And A and T have 2. Pretty nifty. So these are hydrogen bonds. Hydrogen bonds zip up DNA and connect the 2 separate. That's how I would spell it. Separate. Nobody says that. Separate. Come on. Separate. Nobody says separate. English makes no sense. 2 separate strands. Okay. That's why I can't spell, because it makes no dang sense. Okay. So interestingly, let's say you're a little tiny microbe and you live way down in the ocean. So let's say we're way down deep in the ocean, way down here, and there is let's say a thermal vent that's very, very, very hot, and we are a teeny tiny little microbe. Just a little guy. We live way down here. We're just chilling like this. And we got our little DNA strands like this, right? If you look at our DNA, microbes in hot environments will have high GC content in their DNA sequence. Sequence, I know I spelled that wrong. Sequence. Because GC has 3 hydrogen bonds. So it helps stabilize the DNA in high temps. So the 2 strands don't come apart. Stabilize hydrogen. Okay, I'm going to learn how to spell by teaching. Pretty nifty, right? That a tiny little microbe like that will have higher GC content because it has 3 hydrogen bonds. It's almost like evolution is true. Very fascinating. All right, one little thing. This deoxyribonucleic acid means that there's actually an oxygen missing off this sugar, an OH group, and hydroxyl group actually. And so that makes DNA way more stable. If that oxygen is present, sometimes that can interact with the phosphate below and kind of break this chain down. So it'll break the chain, and that's what happens in RNA. It's not missing that hydroxyl group. So what will actually happen with RNA is it will degrade itself. So the hydroxyl group in the sugar will interact with the backbone and break that strand. And then this strand will break here. And so over time, RNA degrades very quickly. RNA degrades very quickly. That's what I want you to remember. I don't need you to know the biochemistry of it, but RNA degrades very quickly, like on the span of a few hours to a day at room temp. RNA breaks down, something like that. Whereas DNA is stable for like 10,000 years or more, depending on the conditions. Pretty crazy. One little biochemical change causes that difference in stability. Okay. One last little thing I want to talk about is ATP, which is the energy currency of our cells. So if we just draw this guy out here, again, I'm just going to bring him down here. So what we do is we take sugar and we break it down and we get energy out of it. And eventually, and we'll talk about how we do this, we make ATP. And ATP is essentially just this. It's a nucleotide with 3 phosphates instead of just 1. That's ATP. So we break down sugar. We break down sugar to fuel the production of ATP. Production sugar. Okay. And ATP is very good because it's a very nice little energy source. So this bond here, chemically, this bond has a lot of energy in it. Very energetic. So when we break this bond, copy these bad boys over here. When we break it, this bond's broken. What happens is it's like an explosion. An explosive amount of energy is released when the bond between the bond of the third phosphate group is broken. Phosphate. Pretty nifty. Pretty nifty indeed. So we use this as an energy currency. So now that's depleted. So then what we can do is, through a process of breaking down more sugar and extracting the energy from that, we can then make more ATP. And something that this would be useful for, fueling, ATP, for example, would bind to this enzyme, maybe on its butt right there. And it would release all that energy to cause this shape change to smush these two sugars together to make a polymer. That's just one thing that ATP is used for. Copy DNA to pump things across the cell membrane. ATP is how it's the fuel source of cells. Yeah, I think that's all I wanted to cover about nucleotides. There's a lot. I guess I'll just pull this up. It's pretty cool. So we can just kind of see this together. You don't have to draw this out. But just to kind of see these interactions. So we got our phosphate groups on the back. We got our sugar. And the two strands of DNA are actually, it's called anti-parallel. They're going in opposite directions. But you can see how we have these 2 hydrogen bonds between A and T, 3 between C and G, so on and so forth. So DNA strands are held together. They interact with each other through hydrogen bonds, which is just like a little weak magnetic attraction. All right. that was a lot more than I expected to kind of have to go through with this, but that's all really important stuff for you to understand how cells work. We'll save cells for next week. I hope you learned something. Let's see. Let's go back to our original. I think I covered everything. Organic molecules, molecular molecules, biomolecules, promises of this topic. You'll better understand your food. What is that stuff that I'm eating? Nutrition labels will make more sense. You'll be better at cooking, and you'll understand the chemistry that you're made of, and you'll understand why, like, not just meat is protein. Like, cows eat grass, and look at them. They're made out of meat that we eat, and they just eat grass. It's pretty cool. It's pretty cool indeed. All right. I'll see you guys in the discussion post and in lab.